Hey, good morning. Are there any questions before we begin today? Okay, so today we're going to be talking about the lymphatic system. So your learning outcomes are to describe the structure and function of the lymphatic system, explain the origins and components of lymph, and explain how the lymphatic system interacts with the circulatory system. So the lymphatic system consists of lymphatic vessels and organs, and there's four main functions. Lymphatic capillaries absorb excess interstitial fluid and return it to the bloodstream. Lymphatic capillaries, called lacteals, in the small intestines absorb dietary fats and transport them to the bloodstream. And then production, maintenance, and distribution of lymphatic sites in the body. And this also helps in defense against pathogens. So lymphatic vessels carry a fluid called lymph and begin as blind-ended lymphatic capillaries in the tissues. As lymph moves away from the tissues, the capillaries merge into lymphatic vessels and then two lymphatic ducts, the thoracic duct and the right lymphatic duct. The larger thoracic duct collects lymph from the body below the thorax, the left arm and the left side of the head and neck, and empties into the left subclavian vein. The right lymphatic duct returns lymph from the right arm and right side of the head and neck into the right subclavian vein. So that's illustrated here. So we have the right lymphatic duct, which empties lymph into this right subclavian vein. We have the tonsil, which is a patches of lymphatic tissue that helps to prevent entrance of pathogens by way of nose and mouth. We have red bone marrow, the site for the origin of all types of blood cells. We have the thymus, which is a lymphatic tissue where T lymphocytes mature and learn how to tell self from non-self. We have the spleen, which cleanses the body of cellular debris and bacteria, while resident lymphocytes respond to the presence of antigens. Then we have the thoracic duct, which empties lymph into the left subclavian vein, and the axillary lymph nodes located under the arm region. So lymphatic vessels continued. The anatomy of the larger lymphatic vessel is similar to that of cardiovascular veins, including the presence of valves. So valves prevent lymph from flowing backwards. The movement of lymph is largely dependent on skeletal muscle contraction. So there's two types of lymphatic organs. There's primary and secondary. So primary lymphatic organs are red bone marrow and the thymus. And secondary lymphatic organs are lymph nodes, the spleen, and the tonsils. And that's shown here. So tissue samples from primary and secondary lymphatic organs. So on the left here, we have red bone marrow, the thymus, the spleen, and lymph nodes. So red bone marrow is the site of blood cell production. In children, most bones have red marrow, only a few do in adults. And this is where B cells or B lymphocytes mature. So the thymus is bilobed. It's in the thoracic cavity superior to the heart or above the heart. It is largest in children and shrinks as we age. And there's two functions. This produces thymosin, a hormone that induces the maturation of T cells or T lymphocytes. And immature T cells move from the marrow to the thymus where they mature. So secondary lymphatic organs, the spleen filters blood, is in the upper left region of the abdominal cavity. Connective tissue divides it into white pulp and red pulp. Macrophages in red pulp remove pathogens, debris, and worn out red blood cells from the blood. It has a thin outer capsule, so it can rupture from trauma. And then the lymph nodes are found along the lymphatic vessels. They filter lymph. There's connective tissue that forms a capsule around it and divides it into compartments. It's filled with macrophages that engulf pathogens and debris. And it also houses lymphocytes, which fight infections and cancer cells. And they're usually named for their location. So they're common in the neck, armpit, and the groin regions. Lymphatic nodules are concentrations of lymphoid tissue that don't have a capsule. The tonsils are located in the pharynx, have the same function as lymph nodes, fight infections that come in through the nose and mouth. And then these Peyer's patches are found in the intestinal walls and the appendix, and they fight infection that come in via the digestive tract. So moving on to innate immune defenses, your learning outcomes here are to list examples of the body's innate defense, summarize the events in the inflammatory response, and explain the role of the complement system. So the innate immune defense is basically your first attack of immunity. So this is the killing or removing of foreign substances, pathogens, and cancer cells from the body. So there are two branches of our immune system. You have innate and adaptive. So innate is fully functional without previous exposure to a pathogen, and adaptive is initiated when exposed to a pathogen. So innate or non-specific immune defenses include physical and chemical barriers like your skin, the inflammatory response, and protective proteins. And the innate defense has no recognition of a pathogen or no memory. So first line of defense against pathogens. So barriers to entry, you have physical and chemical. So the physical is the skin and the mucous membranes. The skin has a lot of keratin, so it is very tough. And also exfoliation carries microbes away from the body. Mucous membranes also have ciliated cells. The cilia sweep away mucus with entrapped pathogens. And there's also chemical or acidic secretions from the sebaceous glands that weaken, kill bacteria on the skin. And then sweat, saliva, and tears have lysozyme or an antibacterial enzyme. So chemical barriers continued. The acidic pH of the stomach inhibits or kills bacteria. The normal flora, the microbes that usually reside in the mouth and intestine, prevent potential pathogens from taking up residence. However, chronic use of antibiotics can make one susceptible to infection by killing off the normal flora. So that's illustrated here. So you have four responses in your innate defenses. You have barriers to entry, you have protective proteins, you have phagocytes and natural killer cells, and then you have the inflammatory response that can recruit macrophages and neutrophil to the site of infection. So the inflammatory response is the second line of defense against pathogens. It employs many neutrophils and macrophages to engulf pathogens. And there's four hallmark symptoms. So redness, heat, swelling, and pain. So chemicals such as histamine released by damaged tissue cells and mast cells cause the capillaries to dilate and become more permeable. Excess blood flow causes the skin to redden and become warm. Increased temperature inhibits growth of pathogens, and then increased blood flow brings white blood cells to the area. So here's the steps of the inflammatory response. So first, injured tissue cells and mast cells release histamine, which causes capillaries to dilate and increases blood flow. Then macrophages phagocytize pathogens and release cytokines, which stimulate the inflammatory response, which recruits neutrophils and monocytes, which can initially become macrophages, and squeeze through the capillary wall and phagocytize pathogens. And then you have blood clotting that walls off capillaries and prevents any blood loss. So increased permeability of capillaries allows fluids and proteins to escape into the tissues. The blood clot prevents blood loss, and excess fluid in the area or swelling presses on nerve endings, causing pain. So white blood cells move out of the bloodstream into the surrounding tissue. Neutrophils are first. They phagocytize debris, dead cells, and bacteria, and they can usually localize any infection and keep it from spreading. If die off in great quantities, they become a yellow-white substance called pus. If neutrophils become overwhelmed, they secrete cytokines, a chemical that attracts more white blood cells, and monocytes that come to the area become macrophages, which are powerful phagocytes. So protective proteins, like in the complement system, there's several plasma proteins designated by the letter C and a number. These are all involved in amplify the inflammatory response. Some bind to mast cells, triggering histamine release. Others attract phagocytes to the scene. And then what's really cool is some form a membrane attack complex that produces holes in the surface of bacteria where fluids enter the bacteria cell and they burst. So that's shown here, showing you the membrane attack complex. So complement proteins can bind to the pathogen and form this complex, which allows fluids to rush in. And these fluids essentially will burst the pathogen. Okay, protective proteins continued. So we have interferons, so chemicals produced by cells that are infected with viruses as a warning to other cells. They bind to receptors of non-infected cells, causing them to produce substances that interfere with viral replication. Okay, so moving on to adaptive immunity. This comes into play when innate or non-specific defenses have failed to prevent an infection. Some provide protection against cancer. These respond to antigens, or the immune system recognizes it as foreign. So antigens can be fragments of bacteria, viruses, molds, parasitic worm can also be antigenic. So abnormal plasma membrane proteins produced by cancer cells may also be antigens. So the immune system is able to distinguish our cells, from non-self pathogens. So how does this work? So it depends on the action of B cells or B lymphocytes, or T cells or T lymphocytes. So B cells and T cells have specific antigen receptors, which are plasma membrane proteins that bind to particular antigens. And so each lymphocyte has only one type of receptor. The receptor and antigen fit together like a lock and key. So there are two pathways of adaptive immunity. You have cell-mediated and antibody-mediated. In cell-mediated immunity, T cells kill cells that are presenting a specific foreign antigen. In antibody-mediated immunity, also called humoral immunity, B cells produce antibodies that bind to free antigens and body fluids. So here's an overview of adaptive immunity. So again, we have cell-mediated immunity and also antibody-mediated immunity. And both of these work by B cells and T cells that can produce a memory. And when they're not in use, they go through apoptosis, but they also can induce apoptosis in infected cells. And lastly over here is they can produce antibodies. So B cell receptors are protein receptors on the B cells. An antigen binds to the B cell receptor on only one type of B cell. That one B cell then produces copies of itself. This group of identical B cells is called a clone. So B cells become plasma cells and memory B cells. So during clonal expansion, cytokines secreted by helper T cells stimulate B cells to clone. Most of the cloned B cells become plasma cells, which produce large numbers of antibodies that bind to the antigen that initiated this whole process. Some B cells become memory cells, which become active in future encounters of the same antigen. So it confers immunity to that antigen. So that's shown here. So there's tons of different B cells. And then when an antigen binds that B cell, this promotes activation of mitosis essentially. And so you get clonal expansion of that B cell that bound to that antigen. And again, if they're not in use, they can go through apoptosis, and they can also promote apoptosis of infected cells. So after an infection has become overcome, the plasma cells are no longer needed and undergo apoptosis. And remember, apoptosis is just programmed cell death. So the structure of an antibody. So these are Y-shaped proteins with two arms. Each has a heavy, long polypeptide chain and a light, short polypeptide chain. These chains have constant regions located at the trunks of the Y, where the sequence of the amino acids is fixed. Antibodies are classified by the structure of its constant region. So that's illustrated here. So we have this heavy chain and this light chain that form this Y structure. And there's two antigen binding sites. And then here's a more detailed picture of that. So the variable regions form an antigen binding site. Their shape is specific to a particular antigen. The antigen combines with the antibody at the antigen binding site in a lock and key manner. So antibodies may consist of single Y-shaped molecules called monomers, or may be paired together in a dimer. Some are pentamers, or clusters of 5 Y-shaped molecules linked together. So neutralization is when the antibodies coat a virus and toxin completely, so they neutralize them and remove the threat. So the immune complex is a clump of antigens combined with antibodies, and the immune complex attracts white blood cells. So there's different classes of antibodies. We just call them Ig, or immunoglobulins. So we have IgG, IgM, IgA, IgD, IgE. So IgG is the major type in blood. Their smaller numbers are found in lymph and interstitial fluid, but these bind to pathogens and toxins, and they also can cross the placenta from mother to fetus, conferring temporary immune protection in the baby. So it's all summarized. I'm not going to read this for you. You can look it over, but it summarizes where the different antibodies are found and their function. So IgM are pentamers. These are the first antibodies produced by a newborn. They first appear in blood after an infection begins, and they first disappear when the infection is over, and they activate the complement system. So IgA are monomers or dimers containing 2 Y-shaped structures. Main type of antibody found in body secretions, saliva, tears, mucus, and breast milk. They bind to pathogens so they can't reach the bloodstream. IgD is an antigen receptor on immature B cells. IgE prevents parasitic worm infections. IgE can also cause allergic responses. Okay, so monoclonal antibodies. Monoclonal antibodies are all identical and are produced by plasma cells derived from the same B cell. And what's really cool is you can produce these in the lab. And these are used for medical tests and treatments. So here's an example of this is basically called a hybridoma cell, because you take a B cell or a T cell and you fuse it with a cancer cell so that it can indefinitely divide. And so it'll basically make an infinite amount of antibody, and then you can pull that antibody out of the solution, and you can use it for experiments or detections and things like that in the lab. So T cells and cell-mediated immunity. T cell receptors are unique receptors on the surface of each T cell. B cells also have unique receptors, but unlike B cells, T cells are unable to recognize an antigen without help. It must be displayed to them by an antigen presenting cell, or an APC, such as a macrophage. After phagocytizing a pathogen, APCs travel to the T cells in a lymph node or the spleen. So T cells and cell-mediated immunity. APCs break the ingested pathogen apart in a lysosome. A piece of the pathogen is then displayed in the groove of a major histocompatibility complex, or the MHC, protein on the cell surface. The 2 classes of MHC proteins are called MHC1 and MHC2. So T cells and cell-mediated immunity concluded. Human MHC2 proteins are called human leukocyte antigens. They're found in all body cells, and there are 3 general groups of HLAs. There's HLA, B, and DR, each with a number of protein variations. Each person has a unique combination of HELs. However, HELs and identical twins are exactly the same. So clonal expansion, many copies of the activated T cell are produced. Occurs when a macrophage presents an antigen to a T cell that has a specific TCR that will bind this particular antigen. This activates the T cell, causing it to undergo clonal expansion. Some T cells become cytotoxic T cells, and some will become helper T cells. Okay, so that's shown here. If you have an inactive T cell, and then a macrophage will have an MHC, or in this particular example, MHC1, which is a self-antigen, and this presents it to the T cell, and this will cause it to go undergo clonal expansion, where you either have a cytotoxic T cell or a memory T cell. And then again, if they're not in use, they will just go through apoptosis. So cytotoxic T cells have storage vacuoles that contain perforons and enzymes called granzymes. After binding to a virus, infected cell, or tumor cell, it releases perforins, which punch holes in the plasma membrane, forming a pore. Cytotoxic T cells then deliver granzymes into the pore, causing the cell to undergo apoptosis. So this is responsible for cell-mediated immunity. Okay, so that's shown here. So perforins form holes in the target cell, and then granzymes are sort of injected through the hole, and this causes the target cell to undergo apoptosis, the infected cell. And here's a nice electrogram showing that. You have the cytotoxic T cells bound to the target cell that's been infected. Okay, so helper T cells, these secrete cytokines that enhance the response of all types of immune cells. B cells cannot be activated without T cell help. The human immunodeficiency virus, or HIV, which causes AIDS, infects helper T cells, thus inactivating the immune response. So acquired immunity. So your learning outcomes here are to distinguish between active and passive immunity. Recognize the importance of cytokines in immunity. So acquired immunity can be brought about naturally through an infection or artificially through medical intervention. And there are two types of acquired immunity. We have active and passive. So active immunity, the individual's body makes antibodies against a particular antigen. It can happen through natural infection or through immunization involving vaccines, contain antigens from the pathogens or the pathogens themselves. So they're treated so they can no longer cause disease. The first exposure to an antigen produces a primary response, and a second exposure, a secondary response. And this depends on memory lymphocytes and sometimes booster shots. So here's a graph showing you that. So on the x-axis, it's showing you time. And then on the y-axis, it's showing you the plasma antibody concentration. And so you can see after the first exposure to vaccine, there's a little spike in the amount of antibody concentration. And then if you do a second vaccination, you get a huge spike or a secondary response in antibody concentration. And again, this is depending on the memory lymphocytes. Okay, so passive immunity, an individual is given antibodies to combat a disease. Since not produced by the individual's plasma cells, passive immunity is temporary. So that is, for example, in newborn infants, passive immunity to some diseases because IgG antibodies have crossed the placenta from the mother's blood. Breastfeeding also prolongs natural passive immunity because IgG and IgA antibodies are present in mother's milk. You can also receive antibodies by injection, as in a vaccine. So here you're showing some examples of delivery mechanisms of passive immunity. So antibodies like an IgG can cross the placenta. Antibodies IgG and IgA are secreted into breast milk. And then antibodies can be injected by a physician. So cytokines and immunity. Cytokines are signaling molecules produced by T lymphocytes and macrophages and other cells. They regulate white blood cell formation and our function. That is, interferon and interleukins are cytokines and are used in various medical treatments. So hypersensitivity reactions. Explain what causes an allergic reaction. Identify the causes of select autoimmune diseases. So hypersensitivity reactions, when the immune system overreacts and causes harm to the body. So that is allergies, receiving an incompatible blood type, tissue rejection, or autoimmune disease. So allergies are hypersensitivity to allergens, such as pollen, food, or animal hair, which would normally be harmless. So immediate allergic response occurs within seconds of contact with the antigen. And this is caused by IgE antibodies attached to receptors on mast cells and basophils, which release histamine when they bind allergens. When pollen is an allergen, histamine stimulates the mucous membranes of the nose and eyes to release fluid. This is why you have runny nose and watery eyes during hay fever. With asthma, airways constrict, resulting in wheezing. And then food allergies can cause nausea, vomiting, and diarrhea. Another thing with allergies is you can have anaphylactic shock, which is an immediate allergic response. It occurs when the allergen enters the bloodstream and is characterized by a sudden and life-threatening drop in blood pressure due to increased permeability of the capillaries by histamine. So taking epinephrine can counteract this reaction. You can also have a delayed allergic response, which is initiated by memory T cells at the location of contact with the allergen. So an example there is like poison ivy. Okay, so tissue rejection. Rejection of transplanted tissue results because the recipient's immune system recognizes that the transplanted tissue is not self. So cytotoxic T cells attack the transplanted tissue, can be controlled with immunosuppressive drugs and by transplanting organs that have the same MHC proteins in the donor and recipient. Some immunosuppressive drugs act by inhibiting the production of cytokines. Rejection of transplanted tissue continued. So xenotransplantation or transplanting organs from an animal. Some organs can also be grown in a lab. Okay, and then moving on to the last topic here are severe combined immunodeficiency diseases. So both antibody and cell-mediated immunity are inadequate or lacking. This means a minor infection can be fatal, and you can treat this with bone marrow transplant or gene therapy. Acquired immune deficiencies can be caused by infections, chemical exposure, or radiation. Acquired immunodeficiency syndrome, or AIDS, results from infection with the human immunodeficiency virus, or HIV. AIDS patients are more susceptible to infections and have a higher risk of cancer. Okay, so autoimmune diseases, cytotoxic T cells or antibodies attack the body's own cell as if they were foreign, involves both genetic and environmental factors, sometimes follows an infection that is rheumatic fever. Antibodies induced by a streptococcal bacterial infection of the throat also react with the heart muscle, damages the heart muscles and valves. So that is rheumatoid arthritis, antibodies against joints. So that's shown here. So the antibodies are attacking your own hand, your own joints, causing them to swell. Okay, then we have SLE, or systemic lupus erythematosus. It's usually just called lupus. There's various symptoms like facial rash, fever, and joint pain. This causes damage to the central nervous system, heart, and kidneys, can be fatal, and produces high levels of anti-DNA antibodies. So all human cells except red blood cells contain DNA. So the symptoms of lupus can be exhibited throughout the body. Okay, so another one is myasthenia gravis, or MG. Antibodies attach to and interfere with the function of muscles. This results in severe muscle weakness, eventually resulting in death from respiratory failure. And then multiple sclerosis. T cells attack the myelin sheath, covering nerve fibers, causing central nervous system dysfunction, double vision, and muscular weakness. MS may not be an autoimmune disease because a specific antigen has not been identified. And treatment for all of these diseases are drugs designed to decrease the immune response. Okay, so that's it for today's lecture material. Are there any questions before we end?
I have a question.
Yeah.
So we're gonna be doing chapter 5 or chapter 7 for this week?
You're doing chapter 5 in the lab, and I think this is chapter 7 for the text.
Oh, okay.
Yeah, does that make sense? So it's a little off this week. And
Okay.
the next week we should catch up.
So for next week is gonna be chapter.
I think chapter 7, yeah. Yeah, you can look at the syllabus. I can double check as well.
Yeah, because I saw on the syllabus that for this week it was chapter 5, and then for next week chapter 7. So that's why I was kind of confused.
Yeah,
On
we're
which
just
one?
a little behind in the lab.
Oh, okay.
Are there any other questions? All right, you guys have a great day. I'll catch you later.
Thank you.